Skanhal Pesia, Chinisimpiasi, Holekwaski, Uchines Lemti, Holesia, Keeskutski, Tokekun Sutin, Inazemi Krayu, Hluis West, Chinim Skarupen, Echchakles, and Hop, Tilsentaken, Utskelich, Jerry Ujeri, Hluin Popo, Haslemti, Anant Pesia. Good afternoon, everyone. I'm happy for today, and I'm thankful for everything given from the Creator. My name is Inazemi Krayu, and I have lived 43 snows. I grew up in Butte, Montana. My parents are both named Jerry, which to some of you actually might explain a lot. Thank you, everyone.
So today I will be presenting some of our findings from the ongoing research for the control of Butymus umbellatus. It's commonly known as flowering rush. We are doing this by testing two aqueous herbicide solutions. This project is being carried out by us here at the Sailor Scrutiny College Extension Office. It is being funded by the United States Department of Agriculture, the National Institute of Food and Agriculture, and also some funding with the Army Corps of Engineers and Montana State Department of Natural Resource and Conservation.
And of course it is in cooperation with the Confederated Salish and Kootenai tribes. And I thank the Creator for this incredible gift of knowledge that He has bestowed upon me.
I also can't forget to acknowledge Virgil Dupuy. I am here because of all the hard work from him and all the others that came before me. My thesis study really builds on this awesome work that they have done, and I am proud and thankful to have had this opportunity, and I look forward to working with Virgil and the tribes here for many years in the future to keep our lands native and healthy. Alright, so if I was to summarize a thesis statement about my research, I would say that understanding flowering rush's growth potential and invasive qualities are critical in preventing infestations to avoid any further trophic cascades in our Flathead Lake. Flowering rush has proven to be an amazing plant with amazing growth potential, and therefore must be prevented rather than controlled.
So first, I'll give a brief description about flowering rush's life cycle, history, and how it found its way into Montana. Second, I will touch on some of the adverse environmental, economical, and recreational impacts of this aquatic invasive species. And last, I will explain the herbicide treatments being studied to control flowering rush, and the part that I actually have contributed to.
So before the impacts and the control, we must first understand just a little bit about the family of plant that needs our management attention. So flowering rush is not actually a rush. It's in its own family. It really does resemble a large sedge because it has triangular shaped leaves. And so we obviously know this is an invasive aquatic plant because it's invading our local waterways. So we can say that it is from Eurasia, but there are 51 other countries that it is native to, including Africa. So it was first introduced to North America in 1897 through the St. Lawrence Seaway. And then it was first observed in Montana in Peaceful Bay on the northwest shore of Flathead Lake in 1964. It has adapted an awesome phenotype to be able to have both emerged and fully submerged growth forms. And we see both of these life types in the flathead, which compounds the control measures. So only the flower stalk is round actually, and it has umbel-shaped clusters of these really beautiful light pink to rose-colored flowers. And this was one of the reasons why it has spread so prolifically because of its use and ultimately its escape from aquatic gardens. So it has this really starch-filled fleshy rhizome, which is actually the main way that it spreads in our region.
The fleshy rhizomatous root fragments, which are the main population in my study, these rhizomes spread by minor disturbances and spread long distances because they can float on the water currents. And then it really establishes well when it occurs in backwater eddies and sloughs, these low flow areas, right? These docks and near boat launches. It grows prolifically along our lake shores and especially in slow-moving bodies of water. Because of that, flowering rush has become well established in Flathead Lake and spread downstream through the Salish Casanka Dam and to the lower Flathead River and all the way to the Clark Fork River. It has established all the way into Lake Pend Oreille in the Pend Oreille River, and it's now moving into Washington and Oregon, and it's all the way to the McNeary Dam region of the Columbia River. So to summarize my first point, Flathead Lake is a tributary of the headwaters of the Columbia River Basin. Therefore, flowering rush must be controlled or it will infest and damage downstream communities in the near future, affecting culturally important species along the way. So next, I want to show you some examples of the impacts that this aquatic invasive species are having. The one I personally feel should garner the most attention is the impacts to the local food webs, which have already been extremely altered due to the introductions and invasions of so many other aquatic invasives, from large pike to these tiny mysis shrimp ambush territory. And we feel these other invasive species are directly using invasive rush populations for spawning and ambush territory. So of course our native fish habitat is affected, especially where flowering rush forms dense stands in these previously unvegetated areas, just like our study site in the East Bay. You can see from this chart that I stole from a different study, which all of you certainly seen before, because I think each one of you, my instructors, have alluded to this at some point in my study.
So this was a collaborative study done by many organizations, and the lead author was Bonnie Ellis. And this study showed some valuable data about the introductions of invasive species in Flathead Lake and the negative impacts that have happened. So I wanted to use this because it really shows just how extreme those changes have been in Flathead Lake already, which totally reiterates to me just how important keeping these non-native species out really is.
So chart A, as you see here, is from 1915 to 1916. Chart B is from 81 to 83. And chart C was taken from 1996 to 2005. And so basically the size of the species is the number in the population. Notice the 2 times the 2x on the P-mouth chub in the first chart. This one really stood out to me. So that means the population was at least twice as big as represented. So really strong population. Now then notice the third chart at the bottom, the P-mouth chub, I almost couldn't find it. And the lake whitefish has completely started to dominate. We don't even have time to talk about our critical tiny food species, right? But you can see that those pesky little shrimp has gotten rather large in this last chart. So I just wanted to really hammer this point home, that how connected our local food webs can be and how important to keep them intact really is. And it makes it pretty obvious, I think, that the impacts that this invasive species can have on our food web and how they change the predation scale and abundance of our native organisms. Flowering rush supports habitat for the great pond snail. Now I know I've learned this the hard way, right? The great pond snail hosts a parasite that causes swimmer's net. So obviously that affects tourism related to swimming. Plants also interfere with boat propellers, affecting fishing access, thus reducing our recreational opportunities. Here's a picture Alvin took of flowering rush growing prolifically in an irrigation ditch. This reduces water flow and distribution and increases maintenance costs. And then this list, the impacts keep on going, including reduction of property values. We've already talked about impairing boat navigation, which hugely impacts recreational and fishing markets. So this is just a short list, right? But I kind of need to move on to what we're doing to stop the spread. The goal of our research is to develop a herbicide prescription to reduce the abundance of flowering rush by depleting the rhizomes. So the thought is, by applying these herbicides sequentially, annually, over numerous years, it will deplete the reproductive rhizome and ultimately will reduce the impacts throughout the 900-mile Columbia Basin. So there are 2 aquatic herbicides being used. One is called imazapyr and the other is called imazamox. They have both been applied to bare ground during drawdown from the dam in mid-April. And this is approximately 3 weeks before inundation again.
It's actually because of those control efforts and how the dam is actually managed. That is the reason why we can use these 2 herbicides, and they are proven to have very minimal residual. The herbicide is known to bond really tightly to the soil particles.
So I just kind of wanted to build on that and see if the numbers were able to be replicated to give us more data on depleted rhizome and the underground biomass. So then Virgil allowed me to create and generate some hypotheses and begin to collect some samples from the existing plots. And well, this is what happened. I started collecting rhizomes in April 2017, and then again in the same manner in 2018 and 2019. I first built this sampling tool out of PVC. It actually worked pretty good too. It extracts a really good core sample, 6 inches by 10 inches. It's a little hard to clean out, but so I randomly walked in a zigzag on each plot and collected 6 samples from each. Then I bagged the core samples in a 1-pound Ziploc bags, and then I brought them back to campus. So once I was there, I took them into our laboratory, started washing the rhizomes out of the plugs, and then I counted the number of emerging leaves. I weighed the fine root hairs, and I took the weights of our viable rhizomes. So after that initial set of measurements were completed in April, I took those rhizome fragments and the fine root hairs, and I planted them in 4-inch and 6-inch planters and allowed them to grow in the greenhouse throughout the summer. The plants were monitored then in the greenhouse for 4 months, and then I harvested them again and estimated those each again in August and September. I followed these same methods again in 2018 and 2019 so I could replicate my data. I then took this data and I sent it to the University of Montana for some more thorough statistical analysis by some more experienced scientists besides myself. And then I ran a bunch of comparison tests. I ran some chi-square tests, some ANOVA, some t-tests, some z-tests, some f-tests. I kind of just went crazy with asking a bunch of questions. I will discuss those in the results section in just a minute. So I started out with a good, clean sample size of exactly 30 within my simple random sampling method. But to best summarize the years of percent control of the canopy, block 5 was actually removed from the end results. So the block 5 plots were at a lower lake bed elevation with a natural spring that flows right next to it. So it was inundated by water much sooner than the other 4 blocks. So without those 6 samples, I only had a sample size of n equals 24. And so knowing that, the assumptions are that the distribution is normal, as well as that the sample was chosen randomly. So our entire population would be all of the flowering rush in Flathead Lake. So, which we actually have a good idea of the acreage infested because of all the mapping that we've been doing, but knowing the actual population is pretty much impossible. So our capital N population equals unknown. And because of that large size of the population, the sample size was limited to only 90 total core samples, 6 per plot, which we talked a lot about actually needing more samples, but the samples we took a lot of time and money and effort as it was, so we stuck with 90, which I'm kind of glad we did. And then I set the alpha level for this project at 5%. We know this alpha level is not really replicable in real time, but it holds us to a much higher standard, I think, when running our statistical analysis.
So I guess I will just jump right into summarizing the results before I run out of time, huh? So when tests were run in 2017, 2018, and 2019, there was a similar pattern of herbicide reduction of rhizome reserves, and the leaf growth initiation was observed. And then I compared these results to past data, and the percent canopy control by habitat is acceptable to begin using in management efforts. It may not be 100%, but it is close enough to start getting a good handle on the existing infestations. So you can clearly see from this graph that I compiled of all of my data combined that there is no way that I seriously would make a graph like this. I just knew that Dr. Berthelot would be freaking out if he thought I was serious. So sorry, I couldn't help myself. So to get serious again here's an actual bar graph of my 2017 sprout counts and my rhizome weights. So I made a graph like this for each year's data. So taking a step back, actually, the table I used to create this graph will help explain a little bit. So the first thing I did with my data was to basically summarize it and find the average weight and count of each treatment. I wanted to know all measures of center, right? This test, I wanted to know about my standard deviation and the mean the most. So basically I wanted to get an image of my distribution. So, which interestingly, looking at the standard deviation of the habitat treatment in red, in 2017 we see a pretty substantial increase from 2.92 to 16.56 in the amount of sprouts counted from April to August, but you actually see a slight decrease in the rhizome weights. And jumping forward to 2018, this year's data showed a decrease in the standard deviation, both in the sprouts and the rhizome weights. If I had my 2019 data in order and could report it now, it too actually is showing similar decreases in the distance that my sample sizes are falling away from the average. So how to put all of this data together into a visual, pleasing image to really show? Well, I'm doing this now with R. So I've been working on importing all my data to RStudio and really doing some statistical analysis. So hopefully by the end of next quarter, I can really have some good, easy to read graphs for you all. In conclusion, I shared a little bit about the life history of flowering rush. Second, I touched on a few of the environmental and economical impacts. And last, we see that 4 years of annual treatments in the habitat plots, the rhizome buds number averaged 7.9%, and rhizome biomass is 19.48% when I compared them to the controls. And the clearcast herbicide plots have an average rhizome bud count of 32.7%, and the rhizome biomass is 29%. So compared those to the control plots. So this shows that with further applications, we could see very strong chemical control results from the imazapyr herbicide habitat. Although, of course, further study is needed to monitor the impacts of external drivers, such as dissolved oxygen changes and sediment buildups. This is only one step in controlling this amazingly adaptive plant. It must be used in unison with all other management methods available. As so many others, I had no idea of the impacts of flowering rush on our local ecosystem until I came to work for Virgil Dupuy at the Salish Kootenai College Extension Office. And it only took a few days before I was waist deep in the cold waters of the North Flathead River, fighting my way through this flowering rush, setting light traps to catch mackerel invertebrates and fish fry. And I suddenly realized just how far away from our relationship with the earth and the water and our animal sisters and brothers, and how we must renew the passion in this next generation by passing down the knowledge of so many of our elders have gifted to us. Lemnam Special.